Helicase is an enzyme that unwinds the double helix and disrupts the hydrogen bonds between the bases, thus separating DNA into individual strands and creating a replication fork. The unwinding of the helix generates strain further ahead in the chain, so as we go, topoisomerase will break, untwist, and reconnect the DNA, always ahead of the replication fork. With the strands separate, we can begin to copy each one, but the enzyme that copies the strand needs a place to start. So an enzyme called primase will anneal an RNA primer at a specific location to kickstart the replication. This primer is about 5 to 10 nucleotides long. Then another enzyme called DNA polymerase III binds to the primer and begins to generate a whole new complementary strand, adding nucleotides to the new chain that was initiated by the primer. Nucleotides enter the enzyme's active site, and polymerase catalyzes formation of the phosphodiester bond that joins each new nucleotide as it is added to the complementary strand. This process will be different for each strand because polymerase will always add nucleotides to the 3' end of the existing strand, not the 5' end. And the strands are anti-parallel, so the direction of replication must be in opposite directions for the opposing strands. On the leading strand, DNA replication moves along with the replication fork, continuously synthesizing the complementary strand and requiring only the initial primer. But on the lagging strand, polymerase has to go one chunk at a time, as new template is made available. These chunks are called Okazaki fragments, and they are around 100 to 200 nucleotides long. Each one will require its own primer in order for polymerase to bind and copy the new fragment. After each fragment is synthesized, DNA polymerase I will go through and replace the RNA nucleotides from the primer with DNA nucleotides to make sure it's DNA all the way through. Lastly, because polymerase can't join the last nucleotide of one fragment to the first nucleotide of another, a separate enzyme called ligase has to go through and